Hello, everyone. I'd like to give you an overview of what we're doing at Seqera, some of our products, some of our contributions to open source, and our platform, which we call the Modern Biotech Stack. So Seqera itself was established coming out of the CRG in Barcelona. And at the time, Paolo and Evan were working on a language called Nextflow, which you will probably know now is a very popular tool that you can use to write data analysis pipelines. As various features, as I'll introduce a bit later on. And at the time, Nextflow was being used for a particular niche set of analysis. And then since then, it's just rocketed in terms of popularity. Paolo has then gone on and added a ton of other features, which have made it really useful. And so, yeah, it started in an open source environment. It's recently hit sort of 10 years, Nextflow, the anniversary. And Seqera itself was initially formed just to deliver training for Nextflow. And so Paolo and Evan would be globetrotting, providing training for Nextflow and spreading the good word. And since then, the company's evolved quite drastically, in fact, to be able to continue and sustain the support to open source, but also developing our own core set of products that we can license, essentially, to make it a lot easier to start using Nextflow on various different environments, and to also allow us to support all of these open source projects. So in Nextflow itself, if you imagine a pipeline you would need to write for RNA-seq analysis, this pipeline will be composed of a number of steps. So in this fancy tube diagram, you can see that you get some FASTQ files off a sequencer, and then you want to put that through a series of steps. And so you have a pre-processing aspect right at the beginning that does some adapter trimming, as well as some basic QC. Then you have an alignment phase and a post-processing phase. And then ultimately, what you want to do is process this data in a way where you can provide scientists with some information that they can use to analyze their scientific experiment. And whether that's, in this case, gene expression counts or variant calls or whatever it is, depending on the context of the experiment, you need to run these sorts of secondary analysis pipelines in order to attempt to start making sense of biological information. And each one of these steps you can imagine as being distinct and can run in its own container, in this case, as I'll introduce a little bit later on. And ultimately, what Nextflow allows you to do is to stitch together pipelines like this, which, if you write properly, can be shared and run virtually anywhere, all the way from a laptop up to a cloud. Nextflow also has various other benefits, including reproducibility, scalability, and parallelism, and these really make it a lot easier for you to start developing and writing analysis pipelines. So in terms of the core features of Nextflow, it's a language, a bit like a conventional programming language, but essentially it's built on top of Groovy, which itself is built on top of Java. And what this allows you to do, essentially, Nextflow is you can imagine each of those steps that I showed you in the previous diagram being stitched together in a pipeline context because you want to process some data. As part of building this pipeline in Nextflow, you can integrate any conventional programming language, including R, Python, Perl, Bash, and so on. And Nextflow really takes care of a lot of the task dependencies and data flow through the pipeline. So as tasks complete, it then submits and manages the execution of tasks downstream based on dependencies. And this just makes it a lot easier for you to then not really have to worry about where you're running the pipeline, but more about what the pipeline should do. And so it abstracts away a lot of these really complex things that you would need to factor in if you're writing proper pipelines. But not only that, Nextflow has been designed from the very early days on. In fact, it was one of the first sort of languages out here to really hinge on using Docker and Singularity and containerization methods. And where this is important is because when you run a pipeline, you need to write it in a way, ideally, where you can share it with someone else to run. And that's only really made possible if you can also ship the software required to run the pipeline with it as well. And so if I ran a pipeline on my HPC with a particular set of tools like SAM tools or BWA, I need to be able to share this with someone else, maybe a collaborator, for example, on the other side of the pond, who wants to run exactly the same pipeline on a different set of input data, but not have to install all of that software one by one in order to run the pipeline. And so this is really where containers come in. You can have static versions of software installed in containers, and it makes it a lot easier to share and run and make these processes reproducible. And on that point, also using Git version control with Git repositories like GitHub, GitLab, Bitbucket, and so on. Nextflow has sort of enforced this best practice from very early on. And in fact, that's generally how it works. The input to a Nextflow run command is typically a Git repository. And that has sort of, again, changed the way in the way that the scientific community has really thought about how they start developing code and interacting with each other. And it's been refreshing and also really really good in general to see bioinformaticians and others, biologists, coming in and starting to use Git and version control to start writing pipelines, and also maybe even, you know, things like reviewing code and various other things. It makes it a lot more collaborative and easy to track what's going on. But on top of all of these features, Nextflow itself can be run virtually anywhere. And this is one of the biggest benefits, in fact, of using Nextflow, is that, as I said, if you write a pipeline properly, you write the pipeline in a way where you essentially abstract away where it's likely to run. And then you can plug in configuration on top whenever you want to run it on a particular environment, to say, okay, well, today I'd like to run the same pipeline on AWS. Tomorrow I'd like it to run it on Azure. And the day after, I'd like to run it on my laptop. And so using some of these best practices and writing Nextflow, you can run these pipelines virtually anywhere. Nextflow itself has seen massive growth over the past few years. And, you know, right now we're at almost 160,000 monthly downloads of documentation, which is just immense. So people are clearly reading the documentation and are interested in writing their own pipelines, and also maybe learning how to write as well. Okay, so NF Core is a community that was formed very early on alongside Nextflow. In fact, when it was still being developed at the CRG, and so at the time there were some Nextflow community conferences that were happening in a small basement at the CRG. I was lucky enough to sort of join the community and get involved very early on with these conferences. And in fact, they were probably still my favorite conferences to this day. So NF Core actually was formed because a number of people, including Phil Yules, who's now at Seqera Labs and is leading sort of the open source development of at Seqera. So at the time, him and a few others came together and decided that they wanted to create another community alongside Nextflow, which essentially involved having best practice pipelines written in Nextflow. And so this is essentially why it's called NF Core, because it's a lot of the guys that came together at the time were from core facilities. And so NF Core was a way of having a standard set of pipelines that anyone can use and contribute to directly, as essentially a big brain of pipelines, so that you wouldn't need to have the same version of an RNA-seq pipeline in 15 different labs and maintain that and various other things. It made a lot of sense to start using some of the benefits I mentioned with Nextflow to really start sharing and developing some of this best practice around NF Core, so anyone can come and use these pipelines and anyone can come and contribute with GitHub and containerization and various other things. And Nextflow really made a lot of this possible. So NF Core itself, there are various ways that you can contribute. It has its own Slack channel. Nextflow has its own now community discourse as well, and there's a Slack channel for that as well. So there are various communication channels. I'd recommend you go and look at the websites for NF Core and Nextflow to see how you can join these communities. They're very inclusive, and you know, NF Core, as I'll show you in the next slide, has just gone from strength to strength. So yeah, with NF Core, you can contribute in various ways. Phil Yules, who co-founded NF Core, is now senior product manager at Seqera. Geraldine, who some of you may know from the Broad Institute, was doing a lot of good stuff there with GATK and various other things, is now also at Seqera, and is our lead advocate. And I've been involved with NF Core since there were, I think there were 20 or 30 people at the time in NF Core. And I've never really looked back. I fell in love with Nextflow at the time, and NF Core sort of stumbled on my lap. Phil came to visit the Crick at the time, and I was writing an ATAC pipeline, contributed there, and then since gone on to write sort of 8 NF Core pipelines and maintain some of the popular ones like RNA-seq and Fetch NGS. And yeah, it's been really cool. We've built an awesome community there, and the proof is in the pudding, really. As I'll show you in the next slide in terms of growth. But the bread and butter of NF Core is essentially the pipelines. And so here you can see a word cloud of all of the different types of pipelines that we have on NF Core. This is probably a bit outdated now, but we have, I think, 90 pipelines now on NF Core. You can see by GitHub stars, RNA-seq is clearly the favorite, but that's just mostly also because there's a lot of RNA-seq data looking around. A lot of scientists, one of the first experiments they will do is an RNA-seq expression type experiment before they do follow-up experiments. But there are other sorts of pipelines creeping into NF Core. And what's great about this is that you can come into the community and it's completely open source. You can contribute your own pipeline and domain expertise. I may not know about proteomics or metabolomics or whatever else, but we have others coming into the community that bring that domain expertise and want to contribute their pipelines to NF Core so that everyone can use them. And so here in blue, you can see metagenomics pipelines. In red, you can see proteomics pipelines. There's a pan-genome pipeline as well, and there's some image analysis pipelines coming on board as well. And image analysis really does pose different problems to conventional genomics pipelines, including just the fact that the data can be very big when you start processing images, and using something like Nextflow has big advantages to that, because you can't open these images up on a laptop and just manually do things. It takes a lot of resources to actually start interacting with these images, and the more you can streamline in a way that is essentially in a headless mode where you don't have to open an image, the more efficient your processes become. And in fact, the IAM-Cyto pipeline that you can see in orange was something we developed at the Crick Data Challenge quite a while back now, to essentially streamline the analysis of image mass cytometry data. And it's really helped speed up scientific research. So NF Core, as I said, I started very early on, 20-30 people in the community, and now we've just hit 8,000, I think it was last week. And it's just going from strength to strength. We've got more contributors coming in, we've got more hackathons, we've got more people that want to help out with events. This time around, we've got local sites, like we did last year for events and that sort of thing. So we can have hubs for the hackathons all around the world, which is really cool, because you start seeing people around the world really adopting and embracing some of the stuff that we're doing, and also, most importantly, contributing back to see it grow over time and be more useful. There are various resources on NF Core. As I said, we've got over 90 pipelines now. There are now almost 1,000 modules. So this is something that came in with DSL2, which is the second iteration of the Nextflow syntax. And since then on NF Core, it was obviously important for us to start sharing components, because that's what DSL2 brought. It gave you the ability to share and reuse Nextflow components within and across pipelines. And so it really made sense on NF Core for us to start having individual modules for tools like, let's say, FastQC. I mean, I'd run that in RNA-seq, but I'd also want to run that in other genomics pipelines. And so having a single source of truth of that in a repository that we've now called NF Core modules means that we can share this across different pipelines very easily. And we've also built a bunch of tooling on top called NF Core Tools. It's a Python package that we've again maintained from the very early on to allow us to impose best practices in terms of how to write pipelines and various linting requirements and other things. And it's sort of evolved since then to become a lot more comprehensive, including the ability to install, update modules in pipelines and various other things. So I'd recommend you check that out if you haven't already. Okay, so this brings me nicely on to the Seqera platform. So as I mentioned, just to quickly summarize, Nextflow is open source. It will remain open source. It's a very powerful language for you to then start writing your pipelining within a single sort of language, and you don't necessarily have to just write genomics pipelines or omics pipelines. Nextflow can essentially be, you can use Nextflow to write any sort of pipeline that involves containerized software. So, we have people coming to us on NF4, for example, wanting to write astronomy pipelines and earth sciences pipelines as well. So anything you can containerize, you can write a Nextflow pipeline with. NF4 again is another community that sits alongside Nextflow. This is also open source. A lot of the key contributors to NF Core are at Seqera, and we're still pushing the boundaries in terms of how we can make that community better and the pipelines better, and have sort of this feedback cycle with the way that we're adopting Nextflow in NF Core to now ultimately improve it at Seqera for the benefit of everyone. We do have a licensed product called Seqera Platform that essentially where Nextflow is the driving engine behind the Seqera platform. And the platform itself, you can imagine like any conventional sort of user interface, has various features, front-end features, and also other features behind the scenes. And so, as I mentioned, Nextflow is one of the key components of the Seqera platform, but we also have a number of other products, including MultiQC. Now that Phil is at Seqera, we're really pushing the contributions there. We've now hired someone to work full-time on MultiQC to really improve it back into the open source again. MultiQC will also remain open source to improve it for everyone. It's one of the most commonly used bioinformatics reporting tools, and loads of people use it at the end of their pipelines to summarize the results of their particular data. Wave is also another product that we've added, and we recently open sourced that as well. And this essentially makes it, it's a tool that we prefer to make it a lot easier to start interacting and building containers that you need to run Nextflow pipelines. And it has a number of other benefits as well to streamline your interaction with Nextflow and building containers, especially when you start wanting to use private registries and do other more complex things as well. Fusion is something that we will eventually be licensing, and this is something I will go into a bit more detail in a bit. But ultimately, what we want is the Seqera platform to be the modern biotech stack. And so this way, you have all of these core components, the majority of which are open source, and you can run your Nextflow pipelines virtually anywhere using containerization tools that suit you, as well as Git version that suits you, including Bitbucket, GitLab, and various other repositories. And we're building a whole host of other features on top that allow you to do things like interactive data analysis, which I'll go over in a second, as well as trying to automate these processes. So you don't necessarily have to use a user interface. You can also automate a lot of this yourself. So going into these products individually, so Wave, as I mentioned, is a very it's an intelligent way for you to start provisioning and building your containers when you're starting to use Nextflow pipelines. And a very simple example of that is when you write a Nextflow pipeline in NF Core, for example, we have a Conda declaration. So we use Conda wherever we can because it's an awesome tool to ship software essentially. But Conda itself, used standalone, isn't reproducible because Conda needs to have dependency resolution at runtime, and those dependencies can change over time. And so this is really where you need to start using containers. And a very simple application of Wave to solve one of these issues with Conda is that Wave can now use a Conda recipe in a process to then go and build a container that it needs based off that Conda recipe. And behind the scenes, what Wave is doing essentially is using a base micromember image and compiling that Conda environment within the container, so you don't have to manually go and build that. And you can imagine this becomes a lot easier, especially when you want to start having multiple Conda recipes or multiple tools in the same conda recipe being built in a container and not have to manage and maintain that. You change the version in the conda declaration, and you can ask Wave to go and rebuild that, and you don't have to manually build any containers. And so this is just one of the advantages. There are various other advantages that you get, including augmented containers with layers. And actually, Wave was developed partly as a byproduct of this fusion product that I'm going to show you a bit later on, because it allows you to inject the fusion binary into containers, and I'll talk about that in a second. Right. So that brings me on nicely to fusion. And what fusion allows you to do is essentially be smarter with the way that you provision data within containers where Nextflow is running. And so, for example, when you run a typical Nextflow pipeline on an HPC environment, because you're using a shared file system, the data that you need when you're running individual tasks, say, for example, SAM tools or FastQC or whatever else, you can soft link that data from source without having to move that data. And so that's quite efficient because you're not duplicating the data when your pipeline is running. And so the Nextflow will use that, it will process the task, and it will publish the data out. Now, by default, when Nextflow runs in the cloud, that's not the case, because in the cloud, you have blob storage. And so what Nextflow will do by default in the cloud is it will copy the data from a bucket, wherever that data resides, into the virtual machine and the container where the task needs to run. And then it will process the task. If it's a FastQ file, it will run, say, FastQC on it, and then it will publish the HTML results out to another bucket or the same bucket, wherever you want to publish those results. Now, when you start putting more data and more stress on that, and having big FASTA files of human genome references, as well as FastQ files and BAM files, and you're running, you're processing large amounts of data, you can be prone to affecting to getting errors from the cloud vendor APIs. And this isn't necessarily a Nextflow issue or a pipeline issue. It's just the fact that you're moving a lot of data around the cloud. And so to overcome that, what we've done is we've developed this Fusion file system. And what that allows you to do is essentially, rather than copying data from your bucket into your container to run the task, what it does is it mounts that bucket into the container. And it's almost like the files exist directly on in that particular virtual machine, in that container. And so it's a really nice, clever way of reducing the staging in of data when you're running these pipelines, which can have massive impacts and make your pipelines a lot more reliable when you run them. When you use specific instances, say, for example, in AWS, there are some instances and other cloud vendors also that have NVMe disks attached. These essentially come linked with some SSDs that you can use to really speed up I/O operations locally on the disk. And you start seeing much more efficient gains in terms of processing, as well as staging in, and also resilience. So your pipeline ideally will also function for longer without breaking. And so, yeah, this is a really cool sort of product that has matured now over the past year or so since we've been developing it. And we've seen a lot more stability, especially when we're running pipelines on the platform with Nextflow. Multi-QC, as I mentioned, is another piece of that product portfolio. And we're really pushing the boundaries in terms of what multi-QC can do, and coming up with a roadmap itself for multi-QC to make it even better than it is to start reporting and collating and having metrics presented to scientists in a way that they can understand. We have a number of other products that we announced at the Barcelona Summit in October. And so one of this is Data Explorer, and this is essentially like you being able to browse your data directly from within the platform itself. You use the existing credentials you would need to add to the platform anyway in order to perform your compute on your cloud platforms via Nextflow. And then that just allows you to browse this data almost like a file explorer. And there are a number of other key benefits also, in that you can link in public buckets directly, and the platform itself isn't rendering those or actually loading all of that data into the platform. It's essentially just linking to where that data exists to allow you to upload, download, and preview the data that's there. And so it's a really nice way now of also managing credentials and various other things. So when you add users to the platform in a particular workspace, you don't necessarily have to give them independent credentials for your cloud provider for them to access the AWS console and so on. Everything is now streamlined and accessible within one environment. And also another big benefit of this now is that when if there is a failure with your pipeline, and there almost certainly will be, nothing is bulletproof, it allows you to go in and actually troubleshoot your task working directories and various other output files directly from within the platform. You can download the logs and various other things there directly without needing to go and access, especially a cloud vendor type console. So it's streamlining the way that you can troubleshoot and access and upload and play with the platform to get data in and out. There's a number of other features that we'll be building on top of this over the course of the year, but the first couple of iterations for this are, have looked very useful overall. We're also working on an interactive notebook implementation called Data Studios in the platform, and this is also going to be really cool because it now allows you to take these secondary analysis pipelines like the RNA-seq pipeline, which generates, say, for example, differential expression counts, and then plug those into a tertiary analysis environment like the notebook, where you can manipulate the data, you can query the data, you can plot the data and various other things to try and standardize how that looks. And again, all this really is doing behind the scenes is using your existing credentials to provision a container where you can run these interactive notebooks. And behind the scenes, we'll be using things like Fusion to make the storage and the data available as a mount to within the container to make it a lot more efficient to process this sort of data, especially when it's quite large. And it will just make the whole process more efficient to do this. But also, just things like having the ability to use a VS Code container as a data studio, where you can start even hooking into existing cloud resources to start developing your pipelines and that sort of thing. I think that's going to be a really powerful application of this, where you can start, using the platform as a development environment as well. So more to come on this, and I think we'll have some bigger announcements for this. It's currently in private preview, but we'll have bigger announcements soon at the next summit. Right, so the platform itself, there are various ways that you can interact with the platform. I haven't really shown you what the platform itself looks like, and that's quite intentional, because if you do want to see that, then please do feel free to get in touch. There'll be a sort of a contact us page on the Seqera website. Please do get in touch. But overall, there are various ways that you can interact with the platform. The platform itself is a user interface, and that will be the core usage of the platform. However, you don't necessarily need to use the platform in that way. There are various other ways that you can interact with it, including APIs and CLIs. And it's completely up to you how you do that. My preference is to use the command line interface, but that's just because I'm a bioinformatician and I love command line interfaces. But there are other options, including API usage and so on. And so, as I said, you can click a few buttons on the interface and launch an RNA-seq pipeline, or in this case, a fetch NGS pipeline. And sometimes that is what people want to do, including me. There are times where I don't want to interact with the command line. I can quite easily click a few buttons, set up some tests, or run a pipeline with minimal fuss and effort, and it suits my requirements. There's also an API as well, and so the platform itself is composed of three primary components. There's a backend database that stores all the metrics in the application. There's a backend container and a front-end container, and all of the communication between those components is pretty much happening via API calls. And so you can leverage that for your own automation as well. You can hook into existing systems and various other things to try and automate your processes internally to interact with the platform. And in fact, we have some of our big sort of customers using the platform in a high throughput type way, using almost exclusively API interactions. There's also a command line, and behind the scenes, the command line is also making API calls, but it's just doing it and it's presenting a nice sort of command line interface hides behind the scenes all of these different API calls and various other things going on. And so it just gives you a very simple command like launch RNA-seq or have a look at this particular run. And so you install the CLI and you can start doing various things with it to interact with the platform. And again, virtually anything you can do via the user interface, you can do via the command line, like adding users, adding compute environments, adding pipelines, running pipelines, adding secrets, various other things, team management. You can do all of that via the command line if you choose. You can also export entities from the command line, like compute environments where you're going to be running your pipelines, your Nextflow pipelines, as JSON. And so typically the platform itself, we don't own any compute or infra. All of the compute and infrastructure where you would be running Nextflow pipelines is actually still owned by you. So the platform, in the same vein as Nextflow, allows you to hook in your compute and your storage directly into the platform. So it's sort of a pane of glass in terms of where you're running your Nextflow pipelines. And this way, it gives you the flexibility to manage your discounts with your cloud vendors, as well as you managing your infrastructure and storage as you see fit. You don't run any Nextflow pipelines in our environment. And that can become quite important, especially when you start thinking about clinical environments where you want to keep everything firewalled off. And in fact, we have two versions of the platform itself. One is a SaaS type version called Seqera Cloud, and another one is an enterprise version where you can install the application in your own environment. And if data security is a priority for you, that way you can install the application in your own firewalled type of setting. So it becomes hopefully more secure with the way that you're storing and managing your data. And so whenever you create or plug in this compute into the platform, it obviously consists of a core set of settings that you would have used in order to create that resource in them and make it available in the platform. You can export all of that as JSON and store it, and now start not only version controlling your Nextflow pipelines, but also version controlling the way that you are creating these entities within the platform, including compute environments and pipelines and that sort of thing. So you can start version controlling that and streamlining that with the CLI to automate these processes as well. We've also developed a tool called Seqera Kit internally. And so this sits on top of the command line interface. And so all of the typical options you would have exposed by the command line interface are now also available for you to set within a YAML type definition. And it just makes it a lot easier and readable to start now running pipelines or adding configuration or various other things or parameters even directly via a simple YAML file that, again, you can version control if you choose to start interacting with the platform in a nice, simple way. Another advantage of using Seqera Kit is that because it's written in Python, you can essentially import Seqera Kit within the context of Python 2 and start interacting with the typical command line options directly from within Python. So if you have automation set up and you prefer using Python, you can import that functionality to start using the platform command line directly from within Python. And it's clearly up to you how you would like to do that. And hopefully this has shown you the various options that you have to interact with the platform. And so we have a number of customers that are using the platform and also Nextflow and have been for a number of years. And the spectrum, starts all the way from small biotech up to big pharma, as well as academic institutions, the CDC, for example, Moderna, Johnson & Johnson, big pharma, as well as others, commercial vendors like Oxford Nanopore and Element Biosciences. You've got genomics type companies in the UK, for example, Genomics England, who are doing some really good things with sequencing population level data within the country. And so, yeah, the customer base spans all the way from small biotech to large pharma. Anywhere that you can imagine running Nextflow, the platform essentially has a number of features on top, cost optimization, various other things that make it a lot more efficient to start running Nextflow in your environments. And given the fact that we're the home of Nextflow, I think it's safe to say we know how best to run Nextflow in your particular environment, and hopefully we can help you too. So thank you for listening, and hopefully we'll be hearing from you soon. Thank you.